Hello student, welcome to my channel, and today we'll discuss the PG entrance test Botany paper 2013, and we'll get benefits from this previous paper of 2013 MSc entrance test for Botany. So let's start. So here we have our first question that Fries classified algae based on dash. Means here it has been asked us that what is that basis, on which basis Fries has classified the algae. Okay, and so here our answer is pigments. On the basis of pigments, Fries have classified algae. Okay, now move on to second question, and here we have the second question. Surface appendages used by bacterial cells to attach to one another as well as to the host organism are called dash. Okay, so here what is the surface appendages by which a bacterial cell get attached to another bacterial cell? We know we can consider what the process of conjugation. In the conjugation process, two bacteria, first of all two bacteria come nearer, and the bacteria with F plasmid, F plasmid, F factor plasmid, that forms a pili, sex pilus or pili, and by the help of that sex pilus, this is the sex pilus here called as pili or sex pilus singular. So by the help of this sex pili, it attaches to other bacteria. So that is why here our answer would be, what is the answer? That is pili, option A. Clear? Okay. Now question number 3 is a complete assembly of the infectious particles of a virus is known as virion, and the complete set of virion is known as dash. Clear? So what is the question here? A complete assembly of infectious particle of virus means if a virus particle is ready to infect, clear, ready to infect, at that time it is called as virion. Okay, and it is said that the complete set of virion means what are the constituents of the virion? We know virion is a virus and that is ready to attack. Then it is the complete set of what? Nucleic acid plus protein, means nucleocapsid, and protein present in the virus is that is called as what? Capsid. The protein coat is called capsid. Clear? That is why answer would be nucleocapsid. Understood? Now come to question number 4, and here we have the question number 4. For the first time that a virus was crystallized in the year 1935 by, means who crystallized the virus for the first time in the year 1935? For more information, you should know that the first virus, what is the name of the first virus which had been crystallized, and that was TMV virus, tobacco mosaic virus, and that was in the year 1935 by which scientist? W.M. Stanley. Clear? So W.M. Stanley crystallized the virus TMV and in the year 1935. Okay. Next is question number 5. Architecturally, a blue-green algal thallus with which have frequent false branches. Okay, means here you will have to find that algae which shows the false branching, not true branching. Clear? And here our answer is Cytonema. Clear? Because when we observe the cytonema under the electron microscope, we can see that many cytonema filament, they will be present very close to each other. But when we see through our naked eye, means without any microscope, at that time we look the branching pattern. We think that that is the branching, but that is not the branching. Two different cytonema have been jointed each other, and they are present very close to each other, and that is why that look like the branching. So it is the false branching, pseudo branching. Clear? So our answer would be cytonema. Clear? Okay. Now question number 6. Yeast is an important source of what? Yeast is that is an unicellular fungus, and this yeast is very important, very very important in wine industry, in bread industry. And here for this question, it is very very important source of riboflavin. Okay, riboflavin, which is vitamin B. Okay, now come to question number 7. So what is the question number 7? Here we have pseudoelaters found in dash. Okay, say here we have 4 options: Mercansia, Riccia, Funaria and the Sphagnum. You'll have to find out that plant which doesn't have any elater, but it has pseudoelaters, false elaters. Clear? So what would be the answer here? Actually here there is no answer here in this question. For the pseudoelater, answer would be Anthocerus. Question is wrong here. Anthocerus has the pseudoelaters. Okay. And in case of Bryopsida, who are Bryopsida, under Bryopsida, Funaria, Sphagnum, these do not have elaters. But yes, Riccia is also devoid of elaters. But only the elaters have elaters present in Mercansia. Understood what I said? This question is wrong. It should be modified. Clear? This is wrong. Means question has been asked as the pseudoelaters, but there is no any option of the answer. Answer B. You should know. Keep in mind, as a student, you'll have to keep in mind that pseudoelaters are always present in hornwort. Hornwort, example, Anthocerus. Clear? These are present in Anthocerus, but okay, and in Funaria, Riccia, Sphagnum, no elaters found. Clear? So here answer question should be modified, that elaters found in. Okay, here there would be no pseudo. So our question would be elaters found in dash. Answer is Mercansia. Clear? In the Mercansia, elaters are present in the sporogonium. And what is the function of the elater? Elater helps in the spore dispersal. Clear? Elaters are hygroscopic in nature, and they help in spore dispersal. Clear? Let us move on to the question number 8. Question number 8. And what is the question here? Specialized organ of the sporophyte for attachment to the gametophyte is called dash. Clear? So here you'll have to answer that. What is that organ which connects a sporophyte with the gametophyte? What is that? Answer is food. Clear? We know very well that if this is the main plant body and this is sporogonium, this connection point, the connection point is called as the foot, and this stalk-like structure is called as seta, and this is called as the capsule. Clear? Here this is the what? Sporophyte. Capsule is the sporophyte. Capsule, seta, all are the parts of the sporophyte. Clear? So foot is that structure. Here the main plant body is what? Gametophyte. Here the sporophyte is attached to the gametophyte by the help of foot. Clear? Okay. So let us move on to the question number 9. Here we have question number 9. And what is the question number 9 saying? Bryophytes spores are. Okay. Here asking that type of flagella present in the spores in the bryophyte kingdom, the bryophyte class of the plant kingdom. Okay. So you should know that this is a characteristic feature, very important characteristic feature in case of bryophyte, that what happens here? Bryophytes have always two flagella, okay, in their spores, and that is why they are what? Biflagellated in nature. Clear? Keep in mind. Okay. So bryophytes are always biflagellated in nature. Another question. In mosses, stomata appears on dash. Okay. In mosses, stomata appears on dash. And the answer is, that is the capsule. Answer is the capsule. In case of mosses, stomata can be found on capsule. Okay, and you know that stomata are essential for the gaseous exchange. Clear? Next. Come to the question number 11. And what is the question 11 saying? Question number 11 is saying that nerve cells are found in dash. Okay, where can we find nerve cells? Okay, so nerve cells can be found in case of the bryophyte. That is what type of bryophyte? That is liverwort called riccia. Clear? So in the riccia we can get the nerve cells. Next, move on to another question. Question number 12. Fertile branch of fern is called dash. Okay. Here fertile branch of fern is called dash. Answer is, that is the sporophyll. Sporophyll is the fertile branch of the fern. Now come to question number 13. Sclerids are associated with dash. Sclerids are associated with dash. So our answer is, that is what? Sclerenchyma. Clear? Sclerids are associated with sclerenchyma. That means sclerids are the reduced form of sclerenchyma type of cells. Clear? Sclerids are reduced form of sclerenchyma type of cells. Next come to question number 14. Transverse cells are associated with. Okay. Transverse cells are associated with. So guess the answer. What is the answer here? Transverse cells are associated with phloem. Clear? That is the phloem. Now come to question number 15. So what is the question here? Tetra condition is seen in. Tetra's condition seen in dash. So our answer is going to be, what is that? That is the dicot root. Tetra's condition can be seen in dicot root. Clear? Next, question number 16. Which of the following is associated with secondary growth? Okay, here there are 4 options: cambium, phloem, cortex, xylem. So here you'll have to find that answer, that which is associated with secondary growth, and your answer is, that is cambium. Clear? Cambium is always associated with secondary growth. And what are the cambium? Cork cambium, vascular cambium, which are collectively called as what? Lateral cambium. Okay. Question number 17. Bulliform cells are called as dash. Means, what is the other name of bulliform cells? What is the other name of bulliform cells? That is motor cells. Okay. So let us know that what do you mean by bulliform cells? What are bulliform cells? Actually, on the leaves of, on the upper surface of generally monocot leaves, we can see bubble-shaped epidermal cells, and these are also called as motor cells. Okay, and these cells make the leaf curl during the water stress condition. Clear? During the water stress condition, these cells are, due to water pressure, make the leaf curl, and because of this curling of leaf, due to this action of the bulliform cells, there would be less loss of water, less transpiration. Clear? Understood? So here our answer is motor cells. Clear? Bulliform cells are also called as motor cells. Now, which of the following is an auxin receptor? Okay, here we'll have to find out the auxin receptor. And what is the answer here? Auxin receptor is ABP1. Okay, and what does it mean? Auxin binding protein 1. So clear? So auxin receptor is ABP1, that is auxin binding protein 1. Okay. Our next question is, name the primary acceptor of CO2 in photosynthetic dark reaction. Okay. It is asking about the Kelvin cycle, that what is the first acceptor, primary acceptor of the CO2. And what is that? The answer is RUBP, ribulose 1,5 bisphosphate, or the diphosphate. So our answer is A. Clear? So keep in mind, RUBP, or that is ribulose bisphosphate or diphosphate, that is the photosynthetic dark, the first primary or the primary CO2 acceptor in photosynthetic dark reaction. Clear? Question number 20. The enzyme responsible for conversion of fructose 1,6-diphosphate into triphosphate. Okay, here you'll have to answer that what is that enzyme which converts fructose 1,6-diphosphate into triphosphate. Okay, and here the answer is aldolase. Clear? Aldolase is that enzyme which converts fructose 1,6 diphosphate into a triphosphate. Okay, next question number 21. So what is the question here we have? The question number 21 is the ballast element is dash. Okay, you'll have to find out the ballast element, and here our ballast element each answer is, that is aluminum and silicon, that is answer A. Clear? Ballast element is aluminum and silicon. Come to question number 22. Our question number 22 is target pressure against the cell is due to the presence of dash. Clear? Target pressure against the cell is due to the presence of what? What is that? The answer is your vacuolar shape. What is the answer? That is the vacuolar shape, because we know that when what is here? Listen, if this is the cell, inside the cell what is present? Vacuole present. When water enters into the cell, that accumulate inside the vacuole. Okay, that is the vacuole. So water accumulate inside the vacuole, and when vacuole starts enlarging due to accumulation of water, it exert pressure on the cell. Okay, so that is why our answer is going to be what? Vacuolar shape. Vacuolar shape is the correct answer for this question. Next is question number 23. The chief source of nitrogen for a green plant is. Okay, you'll have to answer that for a green plant, the chief source of nitrogen, what is that? That is the nitrate. Nitrate is the chief source of nitrogen. It means green plants, they can absorb the nitrogen in the form of nitrates, not directly to the free atmospheric nitrogen. Clear? Next is question number 24. What is question number 24 saying? Cohesion theory of ascent of sap was proposed by, cohesion theory, what is that? Cohesion tension theory. Okay, and it is the best accepted theory in the plant physiology, and it was proposed by Dixon and Joly. Clear? Who are the two scientists? Dixon and Joly proposed cohesion tension theory. Clear? Let's move on to question number 25. What is the question number 25 saying? Hypanthodium is seen in. Okay, hypanthodium is a peculiar type of inflorescence, and it can be found in case of what? Generally in case of what? Ficus. So hypanthodium is the inflorescence in the fig, that is the ficus. Good. Next question number 26. Fruit of Punic granatum is dash. Okay, what is the fruit of Punic granatum? You know what is Punic granatum? That is the pomegranate. Okay, pomegranate. Clear? Pomegranate, and the name of the fruit of pomegranate is, that is the balusta. Okay, students has clicked Psychonos. Okay, this is the question paper of a student, and perhaps he has answered Psychonos in the black mark, but no, the actual answer is, that is balusta. Clear? So once again, what is the fruit of Punica granatum? That is the balusta. Now come to question number 27. Very, very important questions, and it has been asked in many university MSc entrance tests. And what is that? And here, obdiplostimonous condition is seen in. Obdiplostimonous condition is seen in. Okay, you'll have to find out that family in which obdiplostimonous condition can be found. And the answer is Rutaceae, that is C. Correct answer. Correct answer is Rutaceae, in which obdiplostimonous condition can be found. Okay. 28. Royal Botanical Garden is located in. Okay. Royal Botanical Garden is located in Q, that is London, that is England. Q, England. Clear? Who are the following associated with numerical taxonomy? Clear? Name the two scientists who are associated with phonetics. Phonetics means what? Numerical taxonomy. And they are Sokal and Smith. Clear? Sokal and Smith are associated with numerical taxonomy, or that is called as the phonetics. Clear? Next. Question number 30. Stratification occurs in. Okay. Where does stratification occur? It means stratification more occur in tropical forest. Our answer is tropical forest. Clear? Next, question number 31. Respiratory roots and viviparous germination are the characteristics of. Okay, so here you'll have to find out that plant groups in which respiratory roots and viviparous germination can be seen. Our answer is, that is halophytes. Halophytes in mangrove plants example. In those plants, we can get respiratory roots called as pneumatophores, and viviparous germination means seeds present in the fruit attached to the plant start germinating. Clear? Next is question 32. What is it saying? Community. What is a community? Okay. The answer is, here 4 answers here we have. You'll have to find the correct answer. Okay. Answer is D. That is a group of independent and interacting population of different species in a given area, in a certain area. Okay. Question number 33. Lichen is the pioneer vegetation on which succession? Okay. Here you'll have to find out the type of succession in which lichens are the pioneer, means initiator of the, they are the initiator of the succession, and they are on which type of succession? That is lithosphere. Okay, the lithosphere. Answer is going to be lithosphere. Next, question number 34. Overlapping region between two ecosystems. Name that region which is the overlapping or interconnecting between the two ecosystems, and the answer is ecotone. Clear? What is the answer? Ecotone. Now come to question number 35. DNA is present in. Very simple question. DNA can be found in nucleus, yes. Chloroplast, yes. Mitochondria, yes. Means all the above. In all cases, DNA can be found. Question number 36. Nucleosome model is related to. Nucleosome model is related to organization of chromosomes, organization of cell membrane, organization of protein, none of the above. So answer is A. That is organization of chromosomes. Question number 37. The process of transcription is involved in. What is the process of transcription? We know the transcription is what? In the central dogma, we know information flow from DNA to RNA, that is transcription, and RNA to protein is called the translation. This is DNA to RNA, that is the transcription. So here formation of DNA, here formation of RNA means information is flowing from DNA to RNA. Means what is that? Formation of RNA from DNA. The answer is A. Question number 38. During epistatic interaction operating among the two loci, what shall be the ratio of F2 offsprings when they exhibit dominant epistasis? Okay, here you'll have to answer the dominant epistasis in F2 generation, and dominant epistasis is, that is 12:3:1. Keep in mind, dominant epistasis 12:3:1 and duplicate dominant epistato is 15:1. Okay, dominant epistatis, that is 12:3:1, but duplicate dominant epistatis is, what is that? That is 15:1. And also you should know recessive epistatis. Recessive epistatis actually 9:3:4, and duplicate recessive epistatis is 9:7. 9:7. Clear? Next, move on to another question. The type of gene mutation involving change from purine to purine and pyrimidine to pyrimidine. Okay. In this case, what is the answer? Same bases are exchanged. Same bases are changed. Purine to purine. A purine is in gene mutation. Purine change to purine. Pyrimidine change to purine. That is transition. The answer is the transition. Okay. And if it is purine to pyrimidine. Purine to pyrimidine or pyrimidine to purine, vice versa. In that case, it will be transversion. But here, in this question, according to the question, the answer is transition. That is pyrine to pyrine, pyrimidine to pyrimidine, and purine to purine. Clear? Okay. Next. Come to question number 40. Male sterility is controlled by. Male sterility is controlled by. Okay. So, what is the answer here? The answer is cytoplasmic gene only, because male sterility, that is due to the genes present in mitochondria and chloroplast, that is cytoplasmic gene only. Next, move on to another question. Ti plasmid present in. Okay. So find the bacteria in which Ti plasmid present. Very, very important, that is Agrobacterium tumefaciens. Ti, tumor inducing. Okay. Because Agrobacterium tumefaciens, it induces tumor, and that is, the disease is called as crown gall disease. Clear? So answer is Agrobacterium tumefaciens. Question 42. Beta glucuronidase, GUS gene is a dash. Beta glucuronidase is a what type of gene? What is that? It is beta. Here 4 options here we have. that is selectable marker, microsatellite, reporter gene. Okay, and our answer is, what is that? That is reporter gene. Come to question number 43. When pollen grains from flowers of one plant are transmitted to the stigma of another plant, what is this? Means here there are two plants involved. Okay. So what would be the answer? Autogamy cannot be, it cannot be, getonogamy cannot be, because getonogamy is involving only the same plant but two different flowers. Autogamy, same flowers. Okay, so answer is going to be chasmogamy. Pure line selection. Pure line can be obtained from, what answer is correct? That is selfing. Selfing. From the selfing we get pure line. Next is preservation of fruits and vegetables by pickling them in concentrated salt solution is based upon, what is that? If we put any vegetable in the concentrated salt, then what will happen? Plasmolysis. Obviously plasmolysis will happen. So answer is plasmolysis. Ion exchange theory was given by, who gave ion exchange theory? Answer is Donan. Transpiration is very high in what cases? Very simple, high temperature. In high temperature cases, that is very high. 48, glycolysis occurs. What is the place where glycolysis occurs? Glycolysis in the cytoplasm. Answer is cytoplasm. Okay. Monohybrid cross. Very simple, 3:1 monohybrid cross. And the last question here we have is, what is the last question? Cell organelles are embedded in. Where are present cell organelles? Answer is protoplasm. Inside the protoplasm, cell organelles are present. So that's all for the day. I think you'll get benefits from this video, and we discussed the MSc entrance test, PGT, PGET, that is PG entrance test, Botany 2013 in this year. Okay, and in our next video we are going to discuss the 2014 MSc entrance test questions. Okay, so that's all for the day. Thank you to all of you. Subscribe my channel and share it to your friends.